Pleasure to be here. I feel like I'm at a bit of a disadvantage after that magnificent talk, because I want to get on to some of the latest data we've been showing, and I think it doesn't make sense unless I frame it in terms of the contextualize this in the earlier recordings that we've been saying, and we basically, I'll present what we're calling a theory. So another disadvantage we have is that it's really a nascent theory and arguably not a theory at all, in the sense that it's really a set of convergent observations and not a explanation of consciousness. On the other hand, I think there's a fair argument to say that if you look at all of the different theories of consciousness, there are very few explanations of consciousness. But nevertheless, this theory that we're calling dendritic integration theory I think is better understood as a framework for understanding the observations that we're looking at the moment. And so what I want to get to is new observations on cortical layer 6B and what we think it's doing and why we think this might relate to conscious processing. And as I was just alluding to, I think this makes no sense without some larger introduction. I'll be talking about I could also say we come from a bottom-up perspective, and I'll be talking about in particular, the pyramidal cells of the cortex. So these big neurons. Did that come out on the? Yeah. So these big neurons that constitute 70 to 80% of all of the neurons of the cerebral cortex. So there's something special about this neuronal type and the way the cortex works. And I'll move on later on in the talk to talking about another set of neurons. And as I say, it only makes sense if we go through in a few steps. And I don't have much time, so unfortunately, this could have been a talk in itself. But I'll just summarize it. So we'll have to come back to these details somewhere again today, or perhaps you can go to the literature if it doesn't make sense at this point. But the point is that through biophysical testing of these pyramidal neurons in vitro, it's relatively easy to show that it has different modes of firing. And you'll all be, I think, familiar with the idea that neurons collect inputs and fire. And in some sense, what we think is going on with the pyramidal neuron is this reflects what happens when input comes to the bottom compartment of these very long neurons that have two collecting zones for inputs, one at the bottom and one at the top. And that if it comes at the bottom, it tends to make these cells fire in a sort of steady, sporadic way. And on the other hand, if input comes to the top of these neurons, it changes the mode of firing. And it does so because these neurons have channels in the top of the neuron that completely dominate the output of these neurons. They're calcium channels. They look like a heart muscle action potential. It's sometimes called a plateau potential because it lasts for 50 to 100 milliseconds and makes the neuron, captures the neuron and makes it start bursting. And the hypothesis that loosely called the dendrite hypothesis. Yeah.
There is something covering your
Oh, am I able to? I can't say stop, can I?
I think about you can't say stop.
Well, what's underneath that dendrite hypothesis? So. It's close.
Well, perfect. I don't know how to fix that.
I'll just tell you what the title is. That's the easy way to say it.
Yeah, you can move it.
Okay.
And for the rest, you'll have to imagine it.
Yes. Well, actually, it goes well. So I could say that the one good thing about having a bottom-up theory is that it leaves you with really substantial things that you predict and can test. And for instance, if we're using this kind of an idea as a testable hypothesis and linking it, for instance, to some hypotheses that we just heard about, I think, and perhaps linking it to this difficult problem of what's the difference between memory and imagination and so on. And I guess that I want to say that at a minimum, what we should be saying is that the cortex has two two different pathways. And Nadine alluded to this also in, right at the end of the talk in terms of the laminar separation of this. It seems that there's an internal stream and an external stream, and that's going to somehow be congruent with imagining. And so imagining would be some sort of internal stream, and then vividness or the sensory data would be an external stream. And the hypothesis here is that the pyramidal neuron is able to take these at different parts of the neuron separately and then combine them, and then it completely changes the mode of firing. So let me go back to, I think I can, does that work?
Yeah.
Okay. And the idea is if you extrapolate this to a network level, you can imagine that it's not just that one region is doing this, but the whole cortex is doing this. And so on every level, and now we could be talking about levels in a hierarchy, for instance, each region is doing its local processing. So you can imagine that if you're looking at a tiger, that all of the different, in this case, early features, for instance, are providing actual sensory input or data in the external sense, which would be largely affecting the firing of neurons in a low-level sense. But there's also top-down stream, which could be your imagination, or it could just be some other high-level processing that's coming back down. Could also be high-level thalamic regions, and that's basically capturing a certain subpopulation of these neurons around brain-wide, and that they're firing in a different mode, and they're in some sense they pop out, if you like, just by dint of the fact that those particular elements are matching the external and the internal stream at the same time. And this works at several levels. And so I think the advantage of having this kind of a theory is that we've got predictions about what's going on inside the cell, what's going on at the single cell level, what we predict at the network level, and hopefully this would extrapolate to cognitive claims as well. And also we can do tests on this, and in particular, two-photon imaging, because it's all calcium in the dendrites, and so we can do these in vivo. And I don't have the time to show you all of that, so a decade and a half worth of that kind of recordings. And I want to get to the bottom of the cortex, so let's press on. The other thing that made us feel that we were going in the right direction was a test that we did relatively recently that came out during the pandemic, in which we can also combine this with animal recordings and all the latest optogenetic techniques and so on. And one thing you can do is take is express the channel rhodopsin in the pyramidal neurons and make them excitable. And because now we've got some sort of prediction, it was interesting to ask what happens if you use the light to excite only one compartment of these neurons. We've got just these big pyramidal neurons that are light excitable, but we've got a device for shining light horizontally in the cortex, and we ask the question, what happens to the output of these neurons? And we measured that in a couple of ways that I'll just show one way, which was a so-called juxtacellular patch on one of these pyramidal neurons. So we can see how it's firing, and sure enough, it starts to burst fire. This is the lots of trials of putting the light on the dendrites, and we were surprised to find that under anesthesia it flatlines. And you might think that this isn't so surprising because anesthesia should just suppress firing and so on. But it is really surprising in the context that the light that you put on the dendrite is still hitting the channel rhodopsin and causing the depolarization of these neurons. So we're kind of bypassing the network effect of anesthesia here, and we're looking at a single cell effect of anesthesia. It's actually decoupling, if you like, the effect of the light on the firing of these neurons, and that's got to be something biophysical that's happening at the single neuron level. So we, after testing this in 3 different areas, we're extrapolating with only this much evidence. Let's say we've done sensory cortex in two primary sensory regions and frontal cortex in a mouse, but the hypothesis would be this is doing a cortex-wide decoupling and allowing or preventing the internal stream, the imagination stream, if you like, from being part of the process, and hypothesizing that might well be the lack of conscious perception that we're after in at least one aspect that lack that's due to anesthesia. Yeah.
Have you tried stimulating the bottom part of the neuron and doesn't find that?
Yes, yes, and we can get only under anesthesia actually, but we can reproduce this picture. This picture here, we can put light on the bottom, light on the top, and get this kind of a transformation in vivo. So biophysically, things are going the same way in vivo. Right. And just as I'm still preparing for the actual thing I want to talk about at this point. The other thing that we found was that if we blocked the thalamus, which we know, the higher order thalamus, I should say, which we know projects to what you might think of as the coupling zone in this case. And just before I put that up, this is the projecting fibers from the higher order thalamus in the cortex. We can see it going to layer 1 and to layer 5A. And by the way, I think nobody really knows or could guess why higher order thalamus might go to layer 5A. But there is another sort of set of branches known as the oblique dendrites of the pyramidal neurons, which are also a bit obscure. They're often not talked about. So there's a big collection zone for inputs at the top and a big collection zone at the bottom. And then these straggly dendrites coming out along the apical shaft, so this big trunk of a dendrite, and that's exactly where the higher order thalamus, or one of the places where the higher order thalamus comes in, and we think might be affecting coupling. And sure enough, if we blocked the thalamus, the higher order thalamus, we could also get the uncoupling effect. That's to say, when we put light on the channel rhodopsin expressing pyramidal neurons in vivo, we could suppress what is the original response now at a population level. I don't have time to go into all the details, but if you do a local field potential and not the juxtacellular recording, you now get basically the reflection of a population doing this burst firing. And if you block the thalamus, you can reduce this input, so you can reduce the coupling. And it's not due to anything that's happening in the dendrite, so you can also record the local field potential in the dendrite. So the channel rhodopsin is doing, as claimed, what it was supposed to do in the top of the neuron, but it's not getting so efficiently to the bottom if you block the thalamus. So the thalamus is somehow also part of controlling the coupling of this. And so that all, I should say, just as an aside, that's the anesthetic effect at the population level. We could get very similar effects by blocking certain modulatory receptors. So the metabotropic glutamate receptor blockers had the same effect. So this is red is before, and the other color is after. And so blocking metabotropic glutamate receptors had the same thing, and blocking the muscarinic cholinergic receptors had the same thing. So there's neuromodulation going on this too, and that's also affecting the coupling. And that was kind of interesting for something I also don't really have time to dwell on. I have a talented PhD student who's been working on this in vitro. We can recreate this whole thing in vitro, which is kind of useful because you can start doing really specific things like testing which neuromodulation works and so on. Let's say these are channel rhodopsin expressing pyramidal neurons in vitro. We put the light on the top. We can now do a perfect recording of the cell body, a patch clamp recording, and show the difference. I mean, of course, it's upside down here because it doesn't have the thalamic input. I hesitate to call the slice anesthetized. We don't have an anesthetic in there, although we can put an anesthetic in there. And but even without an anesthetic, they're less coupled. And now let's say you can get more coupling by doing an agonist version of the experiment we did in vivo. So to put an agonist for the same receptors, and you can suddenly cause coupling across these neurons, which makes me feel that we're really chasing something real on at the biophysical level. All this adds up to a theory, as I was saying. First of all, the paper that describes the data I've just been describing is this paper, Suzuki and Larkum. But from that, we now want to include this coupling of the pyramidal neurons in the larger theory that we're calling dendritic integration theory. And it's basically the claim that loss of consciousness occurs if you interfere with any of the various projections or loops that are eventually feeding into this sensitive critical mechanism for coupling these neurons. And if you play around with that, and certainly if you block that cortex wide, then there'll be drastic effects on your ability to perceive the world. Okay, so there's a number of things, claims that entail the dendritic integration theory. I don't have enough time to go through them all, but they're now being itemized in different papers describing both the theory in detail, but also this, for instance, is the Storm et al. Paper looking at lots of different theories of consciousness and what they entail. And actually, the second Bain review included the dendritic integration theory. We've actually, to be honest, we've called it a theory to get it on the table, as it were, so that we can start discussing the observations involved, because I think they are really useful for the sorts of discussions we might have today. It puts a definite framework on what might be going on under the hood. And as I was saying, this back propagation activated calcium spike firing was or backfiring is what we call the detection of the two streams of information at the biophysical level by a single cell. I haven't been able to show you the experiments that show that it really is related to perception, but we did our own version of a perceptual detection test varying the threshold and could literally get the animals to quote unquote hallucinate the signal by potentiating the tuft, and it would lick like it would only lick in other circumstances if there was the stimulus present, and here, even when it wasn't present, we could get it to lick. We can show in various ways, and I've shown you one, that it's dependent on subcortical projections, and in particular, we're talking here, I think, about the thalamocortical axis. That there's something really special about the thalamocortical axis for perception, and that anesthesia decouples this. I won't have time to show you the memory version of this, unfortunately. Which is also interesting, particularly if we're talking about what the difference between imagery and working memory might be, and so on. But there's a strong link that we could get back to if you want to delve into that. And now I want to talk about the bottom of the cortex. It's finally, I think, makes sense to that whirlwind introduction. So what am I talking about? I'm talking about these neurons that are very often not referred to, and in the so-called layer 6B, right at the bottom of the cortex. It's a layer that is kind of obscure. It's hard to record from. Not many people even know about it. It's sometimes called layer 7. In fact, Cajal labeled it 7 for what it's worth. It's been known about since the 19th century anatomists started looking at the cortex, but nobody really knew what to do with it. And it's really come alive in recently because of transgenic animals that made it possible to actually look at it. It's interesting for lots of reasons, and I knew about what I knew about this layer, which we have known as in the field for 25 years or so, is that these neurons, and as unlikely as it sounds, project all the way to layer 1, and that's been known for some time. And I always thought that anything that projects to layer 1 is interesting, I think, in the context that we want to know what those inputs that could be interpreted as the internal stream are doing to pyramidal neurons. I have a really gifted postdoc, Tim Zolnick, who, by the way, is on the job market. If anybody, he wants to run his own lab, which I can only say would be an extraordinary lab when he finally gets to run his own lab. But in my lab, he's been doing equally extraordinary experiments, really meticulous specifically looking at layer 6B, and this came out of some early observations while he was looking at something else, and he showed me these fluorescent neurons at the bottom of the cortex that he'd managed to capture, and I said, that's we finally could start working on this, and so then he made it his mission to know everything there was to know about these neurons, and it's largely come out in two papers. One is looking at the input and one is looking at the output. But what really makes this layer special is that, well, one of the things that makes this layer special is that they have every neuromodulatory receptor known to man. Well, not quite, but it's really a lot, and certainly all of the common ones that you'd be thinking of, including, by the way, the serotonergic receptors, which has this picture here is actually a look at the 5-HT2A receptors, which are being expressed both in the big layer 5 cells and in the layer 6B cells. But that's just amongst many. It's kind of famous for its expression of the orexin receptor, which is to do with the sleep-wake cycle. So if you don't have orexin receptors, or if there's some disorder of orexin, you'll probably have narcolepsy, meaning you'll just compulsively fall asleep all the time. So orexin excites these neurons and is somehow related to waking up and being aware. But as I say, lots of different receptors are affecting this layer. And as I was also saying, suddenly we're able to look at this in the last few years because there are transgenic lines to look at this. They're slightly different, but quite overlapping as well. And what you're looking at on the left here is the DRD1 line that's expressing a red fluorescent protein, and you can see now the very bright layer 6B, but also the projection of these neurons. And it turns out they don't just go to layer 1, but they also go to 5A, and they also go to the thalamus. So I was really interested because it goes to layer 1, but then I acquired an interest for the fact that the higher order thalamus goes to layer 5A, and then it's astonishing to find that these neurons also go to layer 1 and layer 5A along with the higher order thalamus. And it's not just that they target these layers, but they target the sub-layers of these layers that the higher order thalamus targets. So if you look very closely, you see both for the higher order thalamus, which this is our favorite somatosensory higher order thalamus, posterior medial nucleus in the rodent. This goes to the top of layer 1, layer 1A, as does layer 6B. And as you can see very nicely, the axons from layer 6B also go to layer 5A exclusively, as does higher order thalamus. So it's got exactly the same projections as the projections we already think are very much involved in or are necessary for conscious perception. So if you put this back into this scheme, we're basically saying that this 6B is going in at just the nodes that we need to, which we expect is system-wide and is feeding into the system. Now, so then the next thing I'm going to, for time reasons, well, I'll just note that in passing that kind of not necessary at this point, that the other thing we of course know is that there's one of the things that happens when the system breaks down under anesthesia is that you get only local processing, and you can't get you can't recruit the whole cortex. But I'll come back to that notion. I was referring to the fact that Tim's done two magnificent studies. The first one looked at the inputs to layer 6B, and the upshot of all of this, lots and lots of work to try to figure out what's going on, is that there's almost no thalamic input to layer 6B. The thalamus ends up going in one way or another, whether it's first order thalamus or higher order thalamus, to every other layer. it doesn't go to 6B, or there's only trace amounts of activity from thalamus going to layer 6B. And on the other hand, it's layer 6B is a hub for long-range cortical projections. So it's in particular, it's getting lots of input from the so-called intratelencephalic neurons, the IT neurons, that both layer 2-3 and in particular layer 5A neurons are signaling to layer 6B, and some of them are going long distances and down from prefrontal and also up to prefrontal. And on the other hand, as we were just saying, I showed the output in the sense of the expression, but Tim then went to the trouble of actually looking at these the physiology of these connections. So, as I was just already saying, there's the output of 6B, which mimics the higher order thalamus. So it's kind of like the cortex's thalamus in some sense. And when we're talking about that, it's basically got this thalamocortical loop by the scruff of the neck. It's imposing on all of the nodes, and not just the neurons of the cortex that project to the thalamus, and not just the thalamus to which those neurons project. So, and in the meantime, it's been able to show that in some cases it's the neurons that of the thalamus that these large layer 5 neurons are projecting to, but also the other node of that neuron in the cortex. It's got the top and the bottom, or the top and the close to the bottom node of this neuron. So it really has this loop, and in some sense has all of the wherewithal to be, let's say, jumping on top of the thalamocortical loop. So don't panic. I won't go into this. This is just for completeness. And also to say that there are subpopulations of the layer 5, of the layer 6B neurons, which is also part of this paper. So if you want all the details, you have to go to the paper. But these subpopulations are basically meaning that it's possible. We don't know what actually happens. It's possible for by getting a subpopulation of these neurons to project either to layer 1 or to layer 5A in the thalamus, or as it turns out, there's a subpopulation that connect to the superior colliculus. And all of this is going into, when you combine this with the fact that we know that it's orexin sensitive, and we know that it's going to targets like the superior colliculus and the higher order thalamic loop, this is all speaking to something related to attention. Just before I get to there, though, Tim also looked very closely at the physiological effect of these inputs, and one way to do this was to go back in vitro to recording from the targets of these neurons, and looking at what the activation of the 6B axons does to those cells, which we can do by putting channel rhodopsin in the 6B neurons, putting light wherever we need them to see what that input does, and recording from the neuron of choice. And I was skipping over a whole section here of the paper, which shows that remarkably these cells are not just targeting the layers specifically. The other specificity that they have is that they only hit layer 5 neurons. And so they miss all the tufts of, for instance, the layer 2-3 neurons, which are actually more numerous and very complex in layer 1, and only hits the layer 5 pyramidal neurons. And I can't think of any other way that this could come about than during the development, but that might be my lack of imagination. But these are early forming elements of the cortex, and then the cortex sort of grows wider, as it were, and the layer 2-3 come in later. And so it's possible that it all gets wired up early on, and 6B is connected to the tufts of the layer 5, and for some reason they don't form any more synapses. So what's happening here is what we expect to be happening in the final analysis, that we put some light on the top of the cortex. We're assuming a subpopulation of the that's the equivalent to activating a subpopulation of the layer 6B neurons, and we can now record from this layer 5 neuron and ask, what does this input do to the tough dendrites, and how does it affect the neuron? And you can see here one trial and all the trials here from a train of lights, so light pulses. So it's 10 light pulses after another in a fast temporal sequence, and we're cutting off actually the action potentials because we're focusing on the things that are causing the action potentials. Another thing I just didn't get time to talk about were all the dynamics that we expect to happen in the tough dendrites. That after years of recording from these neurons, we know that they've got very nonlinear properties. I alluded to one, which was the calcium plateau potential. There's another one that I like to call NMDA bombs or NMDA spikes, where if you get to a certain threshold, there's a local explosive reaction of the cell in just where the NMDA channels can cooperate with each other. And we think is going to be activated by clustered input. And we think that's what's happening here. So we think that the 6B is able to set off these local NMDA bombs. And the evidence for that is if we block NMDA channels, the AP5 here is a specific NMDA channel blocker, it flatlines. As a matter of fact, it's not exactly a flat line. And what's not flat is presumably the ampere component of the input that isn't being blocked by blocking NMDA channels. And so you can see what you might have expected if there hadn't been bombs in the dendrite, something that looks pretty much like a flat line. In other words, the tuft is so far from the soma, electrically speaking, that a large amount of input there has not much effect. But if you give if you endow the tuft with these nonlinearities, you can get completely the opposite effect. So it's totally unexpected, if you like, from an electrical point of view. And that's what the 6B does to these neurons. So it's not just that they're specific, it's not just that they're on all the nodes, it's that they have a huge effect on this unexpectedly large effect that causes burst firing in itself. And we can compare that with what the thalamus does by doing the same experiment, putting channel rhodopsin in the fibers of the higher order thalamus. And it actually has quite a respectable influence, sometimes also causing bombs in the dendrite, but not as consistently as 6B does. And you can see that's also blocked by AP5 or by an NMDA channel blocker. This is all done in the big extratelencephalic pyramidal neurons, which are the ones that exit the cortex and go down to the higher order thalamus and also to spinal cord and so on. But there's also a class of intratelencephalic layer 5 neurons to which layer 6B also projects, and their axons go elsewhere in the cortex and to the striatum, but not really to subcortical, other than striatum, to subcortical areas, and we can compare it there. Here it's sort of, first of all, it's all slightly less in these neurons, but then they have much less of a tuft in layer 1. And second of all, it's flipped in terms of the relative importance of the input from the higher order thalamus to these intratelencephalic layer 5 neurons. But nevertheless, it's much bigger than you would have expected if you block the NMDA receptors, except, again, interestingly, the AMPA pathway of the higher order thalamus on these layer 5 neurons is much more effective than we had expected, and it's not blocked much. I don't have time to go into all of those details, and effectively we've got now a complex situation of possibilities that now includes this other layer of the cortex, layer 6B, and so may the speculation begin. After this, we don't have, I've got maybe a couple of other things that we can say, but basically we're now on the path to trying to understand layer 6B and put it in its put it in context and try to understand how this could be, in some sense, supervening on all of the things we were saying already about the importance of the thalamocortical context. One of the ideas here is that it could be selecting if you imagine this Christmas tree is the cortical column, it could be selecting particular neurons in this column. And if it is, then you could imagine that there's some rhythmic way that it does this. You could also imagine that it's just picking them like a puppeteer and trying to get them, or that it's and this is Tim's favorite hypothesis, I think, that it's got selective ensembles here, and you can we just don't know, but we have the wherewithal to check this now. So this is where we're going forwards. Let me, in the interest of time, just skip that now. I'll say in the end, we expect it to have something to do with attention, and then to the extent to which this attention will become a statement about consciousness is also going to be an interesting way that this transformed. It's certainly involved in modifying our expectations, literally, both from a scientific perspective in terms of what we predict, and I presume in actual fact it's going on in your brain right now. 6B is affecting your expectations, we would say as well, and your ability to focus on one thing or another. One thing that we could do, initial experiment, just to show that we're on the right track here, is an in vivo experiment recapitulating the same old experiment where we put channelrhodopsin in these neurons and we can put light on the brain, and now we can put a probe, a multi-electrode probe down the depths of the cortex. In this case, we chose to sleep deprive the animals so that they were in a not so wakeful state or not so aware state, such that when we hit these neurons, we can see a difference. And you can see the light comes on at intervals here, and when it does, the animal that seems to be stationary or sleeping reacts, or apparently reacts, with some movements and so on, and we can measure what that corresponds to. And so basically, in these sleep-deprived animals, they're mostly in these slow oscillations that they're reflective of sleep stage. And when the light comes on, which is this blue part for 1 second, it goes into a high gamma mode in that local region where we're stimulating, which is very much a sign of let's say, awake activity or awareness or so on. What's really weird here is that it only lasts for the period of the blue light, and it basically switches back to the delta straight after that, which is not expected. I think if you woke some animal up and you had it suddenly doing high gamma, you wouldn't expect it to, on a dime, just go back to delta after that. It would have to recover. It would have to go to sleep again, essentially. Whereas this is implying that part of the cortex is in a wakeful oscillatory state, but the animal is still in some slow wave sense, still asleep. And there are other things to say. I'm kind of running out of time, so let me just say the bunch of things that will, in the end, relate to things like working memory, I think, and maybe that could trigger part of the discussions either now or later on. Here, amongst all of the amazing things that Tim's been able to find out about these connections, is that they're also facilitating. So the thalamic input in the thalamocortical loop is depressing, meaning that if you have a train of these things, they weaken. Each subsequent pulse gives you less of a response at the postsynaptic neuron. With 6B, they're facilitating. That's to say that each subsequent pulse gives you a bigger response in the neuron, and in fact, they're not just facilitating in a train of pulses, but they're facilitating the trains of subsequent barrages start at a higher point. And to see this, there's too many lines here, I'm sorry. The dark blue line is the same one as the first attempt, and the second attempt is the purple, and the purple here, and the third attempt is also purple there. So you can see that a subsequent train is also facilitating. This is a kind of augmentation, a facilitation that's going on over time across gaps, So it's also got something to do with something that might be called best seen as a memory, or let's say it's the synapse is remembering that it's gone through this before, and it's got something to do with collecting populations of neurons. And if we extrapolate this now to the idea, imagine that the whole thing's wired up like this, such that we've got the thalamus and 6B now is hitting the same nodes, and so the same nodes of the layer 5 neuron in this loop here, and 6B is somehow in between there. You could imagine all of the cases we were just looking at the single cortical level. You could have some way to coordinate over multiple cortical columns, or you could do, again, this could extrapolate to something more like selecting the columns, or it could be something along the lines of having interlacing groups of neurons. And again, that's something that we've yet to establish. But all of these are possibilities and intriguing things that are testable at the level of neurons in certain situations. And since we're not really psychophysicists, I guess we're lacking the core experiment to do at the cognitive level in an animal. All of this will have to be done in an animal, I guess, to get to that level of detail. But nevertheless, this is what we're. So just to sum up, we've got a situation where we think the thalamocortical loop is really intrinsic. We think we've prima facie evidence to say that it's because of its interaction with these really incredible pyramidal neurons, and that they have biophysical properties that can't be ignored in trying to explain how the thalamocortical loop works. And we're adding now this layer 6B to the story because it's so intimately connected with the thalamocortical loop. And this is something we'd like to refer to as the second thalamus in some sense. It's the cortex's thalamus that doesn't get thalamic input, but does get long-range cortical. So it does get driven by the internal stream, and it can capture those cortical columns. And of course, the thalamus is both. It's both an external and an internal stream, and so there's going to be interesting interactions between realness and imagination, and bearing in mind that it also expresses all the serotonergic receptors, for instance, and so on. That there's going to be interesting things that we, in a sense, can't predict. The only thing we predict is it's going to be interesting that will happen when 6B gets activated. And just to finish that whole train of thought, we finally want to find a way to first of all see this from a macroscopic point of view and understand not just the thalamocortical loop, but the way multiple thalamocortical loops integrate and how they integrate with subcortical structures. And so we're in the business now of adding this second thalamus, if you like, to try to understand this at the level of the whole neuron. Now, I have to thank so many people, but in particular Tim Zolnick and so many other people that have contributed to this over the years. Since she's in the audience, Anna Braunig is doing currently the in vitro experiments on the coupling, and is also a really important contribution. But I do feel now that all of these things are somehow converging. Anyway, thanks for your attention.
Thank you very much. We have time for some questions. Yes.
Thanks for the talk. It was fascinating. I was quite convinced by the idea that dendritic integration is very important for distinguishing between processing versus no processing. But to me, the question was more like, is it really distinguishing between unconscious processing and conscious processing? And one way of doing that, for example, would be to show that it distinguishes seeing something in motion versus blindness versus not seeing it. If the populations that encode dot, for example, are decoupled when it becomes unconscious, that would show that it's not just a matter of, processing versus not processing, but really a matter of conscious versus unconscious processing. And motion-induced blindness is particularly interesting in this case because it's also a case where you have a dissociation between attention and consciousness. Because when people attend to the dot, it's going to dissociate faster from consciousness. So you could dissociate both processing, consciousness, and attention by using a paradigm like this one. So, so your prediction would be that you don't have dendritic integration by nuclear finally.
Roughly, yeah. So there'll be some complexity. That's beautiful, and it's exactly in a sense what I see as the way forward for testing this. Course, when we're trying to translate these psychophysical experiments into these experiments, we also have to factor in that we have to be able to do these psychophysics in the animal, and so that will affect which psychophysical experiment ends up being the most practical to use. But so I'm sure that would, so if we could just get inside a human and do all these experiments, I'm sure that would be the best way, or one of the really great things to do. And it may still be, if we can get a good reportable way to examine that in an animal, that'd be great. One of the things that we've been working on in the lab is what we call the air track, or a floating platform system, which allows us to do relatively normal, freely moving behavior, quote unquote, because the animal is head fixed and it's the platform that's moving. It's floating on a bed of air. But this allows us to do some more creative kinds of psychophysical tests, but at the same time look under the hood with techniques like two-photon imaging or even PatchMap and so on. I feel sure that enough brainstorming would get us to the crucial experiment, and I feel very unsure because I don't have a training in psychology, and I'm sure I'm missing literally thousands of good tests that I ought to know about, that we could talk about whether that's the test that we could do. I actually think that we've got a chance to look at imagery, actually, and that you could also look at, well, in some sense, I think the work that's done by Rosemers and colleagues in Amsterdam is along those lines too, where they have where they do contextual modulation of things. And there's another work of postdoc is actually recently come from that has been looking at occlusion, where they have an image which either has all of the image or is missing a quadrant. And when it's missing the quadrant, that needs to be then imagined, quote unquote, by the animal in order for that to be a stimulus. And so you can do those kinds of experiments and go forwards. But I would love to just sit down and work out what the best one would be.
We have time for.
So in my understanding, that most of the colleagues we saw them in S1, right?
Yeah.
Also some small part
Yeah.
You're basically describing an imaginary area of effect, right? Like with the 60s different areas interacting kind of across the context. And I'm wondering whether it's from a mechanism, or that's for something like thermal consciousness is what could be implemented similar in different areas, or whether the layer properties of these different areas should change, whether 6B is different than PUC and S1 for example.
Yeah, I'm so glad you brought that up. I didn't plant that. This is completely yeah. This is the question in a sense. Is this a ubiquitous description of a principle that we can take from one region to another and assume that biophysics remain the same and outcome are integrated, or is it changing are the details changing? And and of course we've pushed the line that that the biophysical properties are going to be quite similar, and as we go forward, we're testing that now. And we are now looking in prefrontal cortex in rodents and asking these questions. What will certainly change, and I still kind of regret in some sense that we started in sensory regions, because all our terminology in the beginning was to do with feed forward and feedback. And if we go to prefrontal cortex of the rodent, frontal in humans, or at the top of the hierarchy, then all of this, then what we're calling feed forward and feedback doesn't apply. But what I do think is that I'm going to put this in now. It's not quite the slide I would have picked, but I'm putting it in the last minute. So I was sort of hoping that somebody would ask this question. What I think we should relabel this, it could have gone internal, external, I suppose, but even that doesn't work very well for this. And so another way to look at this is that each column is doing some job, and we'll call it processing a feature for lack of a better terminology. But let's say it's doing some job that is intrinsic to that area. So if it's in primary visual cortex, it's probably doing orientation. But well, we can argue about what it's doing. But the point is that it would be doing something or other, and then you would basically say that the claim would be that what's coming to the bottom of these neurons is feature specific, job specific. Let's say whatever you think that column does is getting the drive at the bottom, and everything else that's not that job, that doesn't pertain to that activity around the brain that is other than that job, is context now for that neuron, as long as it goes to the top of this neuron. So this neuron is now doing what I would say is do I have it? Did I put it somewhere? I didn't write it down actually. I think the job or the operation this neuron or this column is doing now, in terms of the output of the column, is judging the probability of that feature given the context. It's a Bayesian operation, if you like, on that. And it kind of works nicely that this backfiring principle becomes the likelihood problem, the likelihood function in this sense, because it's now the converse. It's the probability of the context given the feature, and the likelihood. I don't want to get too in the weeds here, but the likelihood would be the difficult thing to compute, and would be being done at the single cell level. It would also nicely match the fact that we're not very good at computing likelihood, and we're not very good at Bayesian. I mean, if you just ask somebody to think in a Bayesian way, they don't do a good job. In fact, I think it's not very explicit that we work Bayesian in a Bayesian sense. But it could be possible that that's because it's sort of all built into. You only get one output from this neuron, which is putatively the computation of I do have the probability of the feature given the context. You can only get that out of this neuron. You can't get the likelihood function, which would be up here computed as the probability of the context given the feature. You never get that back, if you like, but you do use it. And then the idea would be that all of these other functions, if you like, are affecting that. And our more recent work has been in anterior lateral medial cortex of mice, namely prefrontal cortex of mice, or frontal cortex as well. And here we've got rule switches, and we're to cut a long story short, we are finding that one way of interpreting what's going on in frontal cortex is it's computing the probability of the decision that needs to be made given the context. And in this context, excuse the pun, is the context here is the instruction that you give it. So it learned some rule. That means in our particular case, we were doing whisker signaling, and we think to go that right. So you could say this neuron or column is computing the probability that it should go left if it gets a left instruction. And sure enough, anatomically, the whisker information from the sensory cortex is coming to layer 1, which makes perfect sense of this construction of the problem. And getting back to the original question, that in the larger scheme of things, of course, you've got everything in the middle now as well. So you've got this delicious situation where at the extreme you might be doing the probability of the decision given the context. And at the other extreme, the probability of the whisker stimulus being a true right stimulus given the context. And you've got every grade of that question in between, and you're doing a massively parallel operation on all of those probabilities, and coming out with, in the end, vividness could be in some sense, I hesitate to call this a prediction, but vividness in some sense would be the return loop of a quote-unquote prediction by your frontal cortex that you ought to be expecting such and such, sending that message to sensory cortex and getting a return message in the form of a different form of firing, a qualitatively different response from those neurons. And you could there, if that all fits, then I would rather say that rather that the frontal cortex is making a decision, quote unquote, it's making a hypothesis. It's basically saying, what if I go left? What if I go right? And then it asks the rest of its brain if that fits, if it gets the congruent response from this, which is kind of like the experiment you're doing.
So we have time for one last quick question. Make sure we have enough time for eating. You will have a comeback.
I'm just trying to wrap my head around this. This is just fascinating. Are you basically saying it's the same mechanism, but because the features change, and the feature can be a decision, the feature can be or the idea of a decision, the idea of a motor plan. That is why kind of the areas are somewhat different ways, but the mechanisms between areas are kind of similar, so that it's almost like not therapy anymore, more like a multiple roles interacting.
Yeah. I wish you could do all my lectures so you can ask the right questions. The point would be that the idea of this being a massively parallel phenomenon is that you've got to get out of the idea that something is happening somewhere. That these two component structures are all capable of doing an operation on an internal and an external stream, and in terms of if you had let's say a higher order theory of consciousness where something's got to emerge, let's say in some sense, what you really need is that that there's some larger, if I can, I don't know if I won't go there. So you've got to have some larger phenomenon which would be the global prediction, which would be impossible to wrap your hands around, as it were, and describe. It would be the total expectation of the non-linear response of the system given this pattern or that pattern, if you like, which is now very hard to describe in terms of this region saying this to that, or this region saying this to that, because they're all speaking simultaneously along two streams and collecting the most resonant response of that. I think what one would want to add to that is a way to eliminate near hits, as it were, and there's a lot of our work has been on also on inhibition and the different ways that you get this. And one of the problems I see going forwards is that what we're starting to find is that when you do get activation of a column, it really means, or when we say, in my terms, activation of the column would be activation of these ET pyramidal neurons. And they tend to be a small population. So it's not the whole column that goes to line. In fact, it's the column is actually mostly inhibited by when the time comes, or in our case, when it does the action or it's apparently perceives something. Most of the neurons are being suppressed, and a few neurons are spiking like hell. And in a naive and untrained animal, it's basically much more small at all. There's kind of nothing really over responds or under responds. And suddenly, in a situation where you would guess that it's quote unquote perceiving, you've got a small population of neurons that are going, but they are obviously bursting in some sense that is very superlinear and exceptional. essentially it's a way that these neurons alone pop out. So you're getting that's another way of putting it, you're getting a pop out phenomenon without a homunculus, so that nobody's saying you, or maybe 6B to some extent is controlling which columns are able to be part of the pop out process, but the pop out is happening in literally intrinsically from the overlap of the internal and the external streams, and over which no one's got control, if you like. That it's going to control itself, isn't it? You're frowning deeply there.